Hi, sorry. My name is Tanya Murkiep. I'm Project Manager with the Alliance Coordinating Office. On behalf of the Canadian HIV Vaccine Initiative Research and Development Alliance Coordinating Office, I'd like to welcome you to our webinar, Do We Need a New Paradigm for HIV Vaccine Development, with Dr. Jose Esparza, formerly of the Bill and Melinda Gates Foundation, which was originally planned as a presentation for our annual meeting in St. John's earlier this year, but due to a delayed flight, Dr. Esparza was unable to attend the meeting and present his talk. So we are very appreciative that he's made the time to join us today. For those audience members who aren't familiar with us here at the CHVI Research and Development Alliance Coordinating Office, we were established by the Government of Canada and the Bill and Melinda Gates Foundation in November 2011 at the International Centre for Infectious Diseases, which is a not-for-profit, non-governmental organization based in Winnipeg. And you can find out more information about us and our partners at our website, alliance-aco.ca. This webinar, entitled Do We Need a New Paradigm for HIV Vaccine Development, will provide Canadian researchers and members of the Canadian HIV Vaccine Initiative Research and Development Alliance with a greater understanding of some of the lessons learned after 30 years of HIV vaccine development. This webinar will include a brief history of HIV vaccine development, some reflections about why it has been so difficult, and personal recommendations to accelerate the development of an HIV vaccine. The webinar will be of particular interest to all trainees and researchers from various disciplines with an interest in HIV vaccine development, but as always, all are welcome. By the end of the webinar, you'll be able to identify different paradigms that have been explored in the search for an HIV vaccine, describe the RV144 surprise, understand the challenges to exploring new paradigms, identify recommendations to accelerate the development of an HIV vaccine, and understand some interesting vaccine concepts. As Elizabeth has said, there will be an opportunity to ask questions during the question and answer period, and you may submit your questions using the comment box that's available at the bottom right side of your screen. And should there happen to be more questions than we can accommodate during the time of the webinar, we will try and get Dr. Esparza to respond to them, and we'll send it out and post it to the participants. So just to get a sense of our audience today, we have a couple of polling questions. The first polling question we'd like to hear from you about is whether you consider yourself an early career investigator, and you can click yes or no, and we'll give you a couple of seconds to respond. Okay, terrific. And another, just for our future planning, we're also curious about how your audio is connected for this webinar. Are you using your computer or voice over the internet protocol in the internet, or have you dialed in with your telephone? Great, thank you. So it's my pleasure to introduce you to our speaker, Dr. Jose Esparza, as I said, formerly of the Bill and Melinda Gates Foundation. Dr. Esparza is an adjunct professor at the Professor of Medicine at the Institute of Human Virology, University of Maryland School of Medicine, and he advises various organizations worldwide working on vaccine development. He was also coordinator of the WHO UNAIDS HIV Vaccine Initiative. Dr. Esparza is author of over 180 papers on different aspects of virology, HIV, AIDS, and vaccine development. He has served on numerous national and international scientific advisory committees and boards, and as the interim president of the board of the Global HIV Vaccine Enterprise. He is a member of the National Academy of Medicine of Venezuela and of the Royal Academy of Doctors of Spain. Welcome, Dr. Esparza.
Many thanks, Tanya. Can you hear me okay? Tanya,
yes, I can. Thank you.
So, as Elizabeth or Tanya mentioned, in the next 45 minutes or so, I will summarize the presentation that I was planning to make at the CHVI meeting in last May. Unfortunately, I missed all my flight connections, and I could not be with you at St. John's in Newfoundland. Today, my presentation does not intend to be a technical one. Rather, I will try to discuss at a very high level the need to bring additional innovation to the search for an HIV vaccine. This search has been a long and difficult one. And who knows, maybe one of you participating in this webinar will be the one who will come out with the solution to the difficult challenge. Can I have the next slide, please?
Yeah, Dr. Esparza, sorry, this is Elizabeth. Do you have a headset on, or can you just pull away a little bit from the microphone? There seems to be some static right now.
Okay, what about now?
That's a bit better, thank you.
Okay. I will try. I mean, this is new technology for me, and I'm speaking from home, so I don't have much technical support here at home. So I will divide my presentation in four topics. I will start with some history, and we'll discuss the different paradigms that have guided our HIV vaccine effort for the last 30 years. Then I will comment on the results from the RD144 trial that I believe is well known to most of you. And I will conclude with some general comments on the need to explore new paradigms. Can I have next, please? Next slide. Next slide, please. As you know, AIDS was recognized as a new disease in 1981. Perhaps some of you in the webinar were not born at that time. Within the next 4 years of discovery of describing AIDS, HIV was identified and convincingly established as the cause of AIDS. Those were years of rapid scientific progress. In 1984, HIV was shown to infect chimpanzees, and this animal was used as an early model for vaccine development. The same year, in 1984, we learned how to produce the virus in large amounts in vitro, also making easier the development, or theoretically easier, the development of an HIV vaccine. Now, in 1985, the protein structure of the virus was described, and its genome was cloned and sequenced. We learned about the genes that this virus has. We also started to learn about the genetic variability of HIV, which has been an issue that has remained an obstacle for vaccine development for many years. And in the same year of 1985, the first simian immunodeficiency virus was isolated, and this provides another animal models for vaccine development. The same year, by the way, neutralizing antibodies were discovered. Can I have the next slide? Next. So there was a lot of optimism at that time created by this rapid scientific progress. And in 1984, the U.S. Secretary of Health and Human Services predicted that an AIDS vaccine would be in clinical trials in 2 years. May I have the next slide? Next, please. So as I say, in 1984, the U.S. Secretary of Health, Margaret Heckler, here with Bob Gallo in the slide, announced that within a couple of years, an HIV vaccine will be in clinical trials. Now, this comment has been misinterpreted many times. They didn't promise a vaccine in 2 years. They promised that a vaccine will be ready for testing in approximately 2 years. Next slide. Next. Now, the prediction of Margaret Heckler became a reality when in 1986, the French Moroccan immunologist, Daniel Sagouri, conducted a clinical trial in Zaire, what is now known the Democratic Republic of Congo. And Daniel Sagouri used a prime boost approach very similar to the one used in RD144. I'm talking about 1986. So he primed volunteers with a vaccinia recombinant expressing the envelope of HIV and boosted with GP160, with the protein. However, this trial went nowhere for a number of reasons, too many to be discussed here, but primarily for ethical reasons. May I have the next slide, please? Next. So after these initial trials occurred in 1986, 2 phase 1 trials were conducted in the United States starting in 1988. These trials tested the safety and immunogenicity of the envelope protein of the virus and also of vaccinia vectors expressing the envelope gene and combination of both. Those were the first 2 clinical trials of HIV vaccine in the United States in 1998. Since then, close to 200 clinical trials have been conducted around the world. Most of those trials have been phase 1 trials to assess the safety and immunogenicity of the candidate vaccines. And the slides show the different vaccine concepts that have been tested, including protein alone, adenovectors, pox vectors, or DNA vaccines. You can see that in the slide. Can I have the next slide, please? Those trials have been conducted according to 3 overlapping waves of vaccine paradigms. Now, the first wave, roughly from 1986 to 2003, the paradigm was based on the belief that the induction of neutralizing antibodies would be sufficient to confer protection against HIV. So different envelope antigens, proteins, GP120, GP160, were tested in phase 1 and also phase 2 trials at that time. Now, the second wave, roughly from 1995 to 2006, was based on information that indicated that CTLs were important in the control of HIV infection in persons living with HIV. And this wave then saw a number of vaccines, mostly vectors and DNA. And the third wave is the current wave that we will discuss later on. Can I have the next slide, please? Now, the neutralizing antibody paradigm was basically modeled after the first successful recombinant vaccine was hepatitis B, licensed in 1986, and as many of you know, it was based on the genetically engineered surface protein antigen of hepatitis B. Very soon, the scientific community learned that you could induce neutralizing antibodies using pieces of the envelope protein, not the whole protein. And this led to the first reductionist approach, which used the V3 loop of GP120 as a vaccine antigen. You may remember that the V3 loop was known at that time as the principal neutralization domain of HIV. But, however, it was soon realized that conformational regions in GP120 contain cross-reacting neutralizing epitopes that could be critical for vaccine development. And this led to the development of a number of whole protein envelope-based recombinant vaccines that entered clinical trials. And there was a lot of optimism in 1990 when 2 different groups reported that those envelope-based vaccines protected chimpanzees from HIV infection. Can I have the next slide? Next. Okay. However, the surprise came in 1994 when we learned that the antibodies induced by those vaccines neutralized only laboratory-adapted strains of HIV, the R5 strains, but not the clinical isolates of HIV, which are the epidemiological and clinical important ones, at least in relation to transmission. So at the end of this paradigm came in 2003, when the GP120 vaccines developed by VacGen failed to provide protection in the first 2 efficacy trials of any HIV vaccine that were conducted in North America and Thailand. Now, I have to say that there are new efforts to develop vaccines that induce neutralizing antibodies. And today, we know that about 20% of HIV-infected people develop broadly neutralizing antibodies. But those antibodies arise only after 2 or more years of infection, and they show extreme hypermutation and other characteristics. So this process of affinity maturation is a challenge that we are confronting now through the use of sequential immunization with mutated antigens. And I want to mention 2 names. Bart Haynes from Duke and Dennis Burton from Scripps are pursuing this approach, although none of them have entered clinical trials so far. Can I have the next slide? As I mentioned, the second paradigm that was explored was the induction of CTLs. Can I have? I want to mention something here. That can I have the previous slide, please? The previous slide. So you see the paradigms are not sequential; they are overlapping. So while people are pursuing the neutralizing antibody paradigm, other people are exploring the CTL paradigm, and that overlapping created and has created a lot of controversies between people exploring or defending the different paradigms. Can I have the next slide, please? Next, please. So the CTL paradigm, as I say, was developed after the recognition of the role of CD8 cells responses in the control of HIV infection. And we are all familiar with the work, for instance, with the work with elite controllers. Now, the challenge has been how to translate those observations from elite controllers, from HIV-infected people, to the development of preventive vaccines. In any case, the type of vaccines approaches that were explored to induce CTLs, as you know, were DNA vaccines, adenovirus vectors, and also post-virus vectors. Now, in non-human primates, most of these candidate vaccines fail to protect against acquisition of HIV infection, although those vaccines, or some of those vaccines, actually decrease virus load in vaccinated animals who become infected. Now, this observation led by 2007 to the conclusion that a vaccine capable of achieving sterilizing immunity may not be a realistic goal, and that perhaps the best we could do is to develop disease-modifying vaccines. Can I have the next slide, please? Next. Yeah. Now, in 2007, as some of you may remember, we learned the results of the STEP and the Family trials that use an adenovirus 5 vector developed by Merck. Those results show that the vaccine did not provide protection. Moreover, in some volunteers, and I don't have time to discuss that today, but in some volunteers who received the vaccine show actually an enhanced acquisition of HIV infection. In 2008, NIAID convened a summit to discuss those results, and they recommended to return to basic science. Basically, they hinted that before moving to additional clinical trials, we should learn more about the biology of HIV. Can I have the next slide, please? Then it came the RV144 surprise. In October of 2009, we learned that this regime tested in RV144, which is a prime with ALVAC, the canary pox expressing envelope antigens, and GP120, provided some protection, modest protection, but real protection, 31.2% protection. I say that this was a surprise because the results from this trial were against conventional wisdom. Success was not expected by the scientific community. And you can go back to the literature and find how basically RB144 was ignored by members of the scientific community during the years that it was conducted. Now, the other surprise was that the protection that was observed was against HIV acquisition, not against decrease in virus load. Was actually, this vaccine was induced something like sterilizing immunity that only 1 year before we thought that it was impossible. Now, the immune correlates, and this is beautiful work coordinated by Bart Haynes, show that the potential correlates of immunity were not neutralizing antibodies, but actually antibodies, binding antibodies to the V2 loop, probably ADCC antibodies, antibody-dependent cell cytotoxicity. Now, we need to confirm and to extend these results from RB144, and there is a coordinated global effort that we refer to as the P5 to conduct additional trials of this concept in the Republic of South Africa. Can I have the next slide, please? So the RB144 surprise tells us that we need to be less dogmatic and that we need to search for new paradigms. Now, there are pros and cons in searching new paradigms. In one hand, working without a clearly defined paradigm can create anxiety among scientists who are exploring new frontiers. But on the other hand, working without what we call the tyranny of paradigms gives us the freedom to explore. Now, I give you, because I'm talking to mostly Canadian scientists, one example of a new paradigm, and it's the induction of protective immunity without causing immune activation. And you know, immune activation favors HIV replication. And this is, to some extent, the work of Frank Plomer from Canada points us in that direction. And more recently, a group of French scientists led by Jean-Marie Andréau have reported a vaccination concept that induced tolerance rather than adaptive immunity. And those animals vaccinated with this tolerogenic vaccine actually seems to be solely protected against HIV infection without inducing immune activation or the classical humoral cell immune responses. I invite you to read the recent papers of Jean-Marie Andréau. Can I have the next slide? Okay, this slide is just to emphasize that efficacy trials have been critical in obtaining answers to our vaccine questions and to change paradigms. In fact, we move from the antibody paradigm represented by the two first tests conducted, the recombinant GP120, VAC-004 and VAC-003. We move from the antibody paradigm to the cell TTL paradigm. And that, when we learned that those trials failed to protect against infection. Likewise, you may remember the STEP and Fan-Billy trial that again failed to protect against infection. And then we actually, at that point, we were without paradigms. Then came the RB144, which is the priming with canary pox and boosting with recombinant GP120. And then we entered a new era of being more open-minded and considering different mechanisms for protection against HIV infection. So, but clinical trials, particularly efficacy trials, are critical in our search for HIV vaccine. Efficacy trial, as my friends in the United States say, the proof is in the pudding, and the pudding is the efficacy trial. Can I have the next slide? So, next, please. Next. Yes. So last year, I was invited to think about the lessons learned after 30 years of HIV vaccine research, and I didn't do it myself. I contacted colleagues who have been working in the field for many years and collected their opinion. So I invite you to read the paper. But basically, there are 5 lessons. One is that the development of an HIV vaccine is one of the most difficult scientific challenges that biomedical sciences is confronted. So we learned that in 30 years. 30 years ago, we thought that the vaccine would be easily developed. Second, the temptation of just following the fashion, meaning the current paradigm, should be avoided. Particularly younger scientists, please be free to explore. Don't be constrained by existing paradigms. The third lesson is that clinical trials, especially large-scale efficacy trials, are critical. We can work for years misguided by theory, by monkeys, but only an efficacy trial will tell us if we are in the right direction. HIV vaccine research requires long-term commitment and funding, and sustainable collaborations are needed, are essential to accelerate the development of an HIV vaccine. Can I have the next slide, please? Next. So people ask me, why has it been so difficult to develop an HIV vaccine? I just listed a few challenges here, and I'm sure you are aware of many of them, but I go through them very quickly. One is the genetic and immunological hypervariability of HIV, which actually means 2 things. One is the geographical variability, just clades, different clades infect different populations around the world. But also in the individual, the genetic variability is the basis for the immune escape. So a vaccine can generate a protective immune response, but only against certain populations of the virus, and they could rapidly escape to this protective immune response. That's a challenge. The second challenge is the lack of an appropriate, meaning predictive animal models, although I have to say that the SIV or sheep macaque model has been very instructive. Now, perhaps the major challenge is that natural immunity is not protective. With most vaccines are developed trying to imitate the protective immune response that is naturally induced by infection that prevents reinfection. Well, that's not the case with HIV. As you know, people infected with HIV develop all sorts of immune responses that are not protective. So here we have to be, in the development of an HIV vaccine, we have to be better than nature, which is not easy. And perhaps our search should be for the development of non-natural immunity. We could have another webinar to discuss non-natural immunity, but I'll leave that with you to discuss. We also need to protect against mucosal transmission. Another challenge is that HIV integrates into the host cell, and the virus targets the cells in the immune system. So we may have a very short window of opportunity in the development of a preventive vaccine. As I discussed before, broadly neutralizing antibodies are rare and are very difficult to induce in the experimental setting or in clinical trials. And another issue that has not been mentioned very often is that the vaccine-induced immune responses, the one that we have seen in RV144 or in many other humans and animal trials, is of very short duration. For some reason, those immune responses wane after 6 months or so from the inoculation. This may require maybe more frequent boosts to maintain the level of immune response that is required to prevent infection, or we may need to know more about the biology of HIV to overcome this short durability of the immune response induced by HIV vaccines. Can I have next slide? Now, to solve the problem, the HIV vaccine challenge, we may need to think outside the box. But we recognize that there are many pressures to think within the box. If your work is is part of the current paradigm, then you are more likely to get grants. You are more likely to have your papers accepted by journals. You will be invited to scientific meetings. And in general, you will be accepted by your colleagues. If you work outside the box, you may have challenges in each one of the three topics mentioned in this slide. It may have the next slide, please. So the question is, what is the box? Now, what I refer as the box is what Thomas Kuhn, who wrote a very influential book entitled The Structure of Scientific Revolutions. The box is what he calls normal science, or within the paradigm science. It is the space where most people feel comfortable, where the results of the experiments are predictable, where grants are obtained and results are accepted for publication. And I have to say that it's also the box of normal science where useful incremental science is produced. Can I have the next slide? Next, please. But Thomas Kuhn proposed another box that he called revolutionary science. This is the out of the paradigm box, which have less defined assumptions, where risks are higher, but also where potential benefits are equally high. And Kuhn proposed that major advances in science occur when we jump the boxes, what he called the paradigm change. But as I mentioned before, many people feel uncomfortable by not being protected by the walls of normal science. Can I have the next slide? Now, revolutionary science has another name given by Clayton Christensen, who is a respected professor of economics in Harvard. He called it disruptive innovation. And disruptive innovation is a new technology that has a serious impact on the status quo and changes the way people have been dealing with something perhaps for decades. And actually, Clayton Christensen provided this advice was to the business community, not scientists. Can I have the next slide? So last year, I read a very inspiring article published in Nature by Dean Simonton, who is an eminent psychologist from the University of California in Davis, and a person who has spent his professional life working on creativity in science. So he wrote, the creative scientists contribute ideas that are original and useful. Now, the scientific genius, however, offers ideas that are original, useful, and surprising. So creative scientists work in the normal science space, while geniuses actually work in the revolutionary science space. Can I have the next slide? Now, Simonton in his paper goes on to say that surprising originality in the natural sciences is a thing of the past, as vast teams of scientists finesse knowledge rather than create disciplines. In the next slide, I have somebody who is closer to our field. Is Maurice Hilleman, probably the greatest vaccinologist of our time. He wrote a few years ago in 1998 that the complexity, redundancy, and other information in modern molecular biology has created an immense problem for the vaccinologists that have to differentiate to hear the tune from the static. He goes on to say that elemental truths are simple and complexity can be defeated. In some respects, the vaccinologist, the practical vaccinologist, may be seen as a navigator in a small canoe with a broken paddle amid a sea of information. Can I have the next slide? It's in this regard that the physicist, the German physicist Max Planck said many years ago that a new scientific truth does not triumph by convincing its opponents and making them see the light, but rather because its opponent eventually die and a new generation of scientists grows up that is familiar with the new paradigm. So it is up to you, the younger scientists, to explore and embrace the new paradigms that could give us the solution to the HIV vaccine challenge. Can I have the next slide, please? Next, please. So this is a diagram that I think is useful to think in terms of HIV vaccines. It's what is called the Kuhn's paradigm change cycle, Kuhn's cycle. It started with normal science, we call current science, or within the paradigm science. Sometimes this normal science has small changes, drifts, model drift. But there was a point with the current paradigm, no matter how much we play with it, entering crisis. The model doesn't provide a solution. That's where we actually have a model revolution that led to a paradigm change. This is generic for Kuhn. Now, let's try to see this in terms of HIV vaccines. Can I have the next slide? Next, please. So maybe the neutralizing antibody paradigm is created the normal, was the first normal science approach to HIV vaccine. There was a small change. Maybe it's not humoral, it's cellular. We have a model drift. But then the model crisis is that neither those vaccines, those develop, the design to induce antibodies or to induce CTLs, actually confer protection. We were ready for a model revolution. Maybe RD144 is the beginning of this new paradigm that would let us to explore with more freedom of mind other approaches to HIV vaccine development. Can I have the next slide, please? So, normal science use the peer review system, which is a great system to preserve the quality of science, but may not be the best approach to promote revolutionary science. The next slide shows what I call the innovation ecosystem. And I will agree that 90% of the effort should be conducted in the normal science area because it's very well based on knowledge in previous experiments, and the solution can come from normal science. But I'm proposing that 10% of our effort should go to explore innovation. Now, or at least out of the paradigm ideas. I'm very careful when I say innovation because every scientist working in any laboratory in the world firmly believe that they are doing innovative research. So I don't want to offend, and perhaps it's better to talk about all out of the paradigm concept rather than innovative concept. But this 10% cannot be guided by the normal peer review system. It requires leadership from funding agencies, require a very astute selection criteria, and it also requires, it's very important, a supportive scientific community. The scientific community should actually endorse those people who are exploring new paradigms. And it's paradoxical that scientists that value innovation are the first, in my experience at least, to oppose any really innovative idea. Can I have the next slide, please? Next, please. Now, an important element in this innovation ecosystem is the end user. Is the ultimate value to society in terms of knowledge or in terms of products, or both. And in our case, the answer is very clear. Our ultimate goal, the ultimate goal of our work is not to produce more papers, is to develop an HIV vaccine for public health use around the world. Now, can I have the next slide? The end user concept was beautifully discussed by Donald Stokes in his book, The Pasteur Quadrant. Stokes described in his books, which I recommend you to read in your free time if you have any, he described 4 quadrants based on the quest for fundamental knowledge or the quest for practical solutions to problems. And you can see that one quadrant that is a quest for fundamental knowledge, but not for any practical solution, is the pure basic research. The example that he used is Niels Bohr, who described the structure of the atom. In the other extreme, you have the search for pure applied research with no pretension of finding new laws of nature. And the prototype there is Edison, who actually created many new inventions, but no new laws in physics. But the other quadrant is the Pasteur Quadrant. The Pasteur Quadrant that combines both the quest for fundamental knowledge and the quest for practical solutions. Can I have the next slide? Next, please. And this is just to emphasize that this is the quadrant where we like to work. Our work on HIV vaccines is both the quest for fundamental information on the biology of HIV and HIV infection, but also the development of new drugs, new diagnostics, and a vaccine. Can I have the next slide, please? Now, I only have 2 more slides. So this graph comes from a modeling project conducted by UNAIDS and IAVI and different collaborators, showing the projected impact of an HIV vaccine. And I don't have a pointer with me. I don't know if I don't know if you see the pointer, but the green line shows what is the future of the epidemic if we just continue doing what we're doing today here, the green line. Then the orange line below is if we actually increase our level of interventions, but not to full extent. Full extent is the purple, the magenta line here at the end. And this is if we basically expand access to existing interventions to all people in the world. Now, this is actually very expensive, and it has been estimated that the implementing the purple line will cost about $24 billion per year only in developing countries. The plot also model what happens if we introduce an HIV vaccine in the year 2025. It's overlapping, in addition to the implementation of further preventive intervention. As you can see in the dotted line, that the effect of a vaccine, in this case, is a vaccine that has a protective efficacy of around 60%. And you can see in this slide that a vaccine actually will be essential in bringing the HIV epidemic under control. Can I have the next slide? The next slide, please. So, to summarize, do we need a new paradigm for HIV vaccine development? Now, the solution could very well come from a better informed approach to the current paradigms. So I don't want to discourage some of you who are actually working on producing new knowledge in the current paradigms. But 2 examples, one of them I already mentioned, is the broadly neutralizing antibodies, the work of Dennis Burton, Parr Haynes, and many others. And who knows, the solution may come from that. I'm also very intrigued by the approach that Dan Baruch from Harvard is using by designing mosaic antigens that can be used to design CTL and antibody-induced vaccines. And Dan Baruch is using adenovirus 26 vectors and basically synthetically designed envelope antigens. And this is done in collaboration with Johnson & Johnson. And clinical trials have been conducted, and animal experiments are absolutely amazing. However, our minds should be open to unexpected or even better surprising results. For instance, the role of non-neutralizing antibodies that was first uncovered, if you want, I mean not first uncovered, but it was brought to an attention by Bart Haynes when he and his collaborators identified the immune correlates of protection. And Galit Alter, is a fellow Canadian, is doing some beautiful work on non-neutralizing antibodies. The other surprise in the field was when Louis Picker showed that his CMV vectors actually induce non-canonical T-cell epitopes, something that was surprising to the scientific community. But in parallel, I think that we should explore novel paradigms systematically. I already mentioned the tolerogenic vaccines by Andreu, but here the list could be very long, and it's up to you to propose those novel paradigms. My last slide actually is a quote from Emile Roux that I heard from Stan Plotkin in his lecture in the Barcelona AIDS Vaccine Conference in October last year. And Emile Roux reminded us that science appears calm and triumphant when it's completed. But science, in the process of being done, is only contradiction and torment, hope and disappointment. Next slide is just to thank you for your attention. I'm ready. We have 15 minutes or 10 minutes for questions. I cannot read the questions in the screen. It's very small for my eyes at least, and maybe Tanya or Elizabeth can convey the questions to me. Thank you very much.
Yeah. Thank you so much, Jose. That was wonderful. Thank you very much. We do have a few questions. We'll open it up to the chat box. We do have quite a long list, and so if you want to use your microphone, bear with us and just use your emoticon there and raise your hand, and we'll get to you. But use the chat box for now. I have a couple of questions that came in during the registration process to get us going. And the first is related to the relevance of HIV-exposed seronegative cohorts in HIV vaccine development. And Jose, do you have any thoughts that you could relay on that?
I knew about this question, so I tried to provide the answer during my presentation. What Frank Plomer has found is that those individuals who seems to be protected from HIV infection, and they are immunoquiescent. They are not immunoactivated, and perhaps this is pointing to the same direction that Jean-Marie Andreu, that we don't want to induce activation, and this is a dilemma that vaccinologists have, because vaccines actually activate the immune system to produce to induce an adaptive immune response. On the other hand, we know from experience and the STEP trial and the family trial shows that activating the immune system has a yin and yang, can be deleterious. So I think that the cohorts of seronegative exposed individuals can tell us a lot about the biology that favors protection versus replication of the HIV. The challenge is how to translate those observations to a practical vaccine.
Thank you, Jose. Another question that came in: if there's anything you wanted to add regarding the role of the WHO and organizations like the Bill and Melinda Gates Foundation or IAVI. Is there anything else you wanted to say further to that?
Yeah. I mean, I didn't mention, but the world actually invests approximately $800 million every year on HIV vaccines research. $800 million. Most of that, I would say that about 85%, maybe 90% of that money comes from the NIH, the US National Institute of Health. Now, the Bill and Melinda Gates Foundation, where I used to work until very recently, contribute with about 10% of the budget. So I believe, and what is important is that we actually focus on different areas. I mean, that not all funding agencies have the same philosophy, the same priority, the same custom base, and funding has to be diversified. So that's why I believe that funding from the CHBI is important, because you may be funding different people, different ideas those that are funded by other organizations. Now, in relation to WHO, I think, I used to work in WHO. WHO has a very important role, particularly by ensuring that trials are conducted in developing countries where the vaccine is most needed, and those trials are conducted to the highest ethical and scientific standards. WHO and UNAIDS, of course.
Okay, and the next question is, what role, if any, can observing the progression of HIV disease help the process of developing a vaccine? And more specifically, does knowing if someone has been infected recently versus not recently present an opportunity to improve vaccine development?
I think that eventually a vaccine, a good vaccine, should induce both antibodies and cell-mediated immunity. Now, I say that because the information we have to date, particularly information coming from Dan Baruch and seems to indicate that antibodies are very important in preventing the acquisition of HIV infection, while CTLs prevent the progression of disease in vaccinated individuals who become infected. So I think that eventually a vaccine will have to incorporate the two elements, prevention of acquisition and prevention of progression. So the study of progression of disease in HIV-infected individuals will tell us what are the functions, the epitopes to target more effectively to prevent the progression to disease. So it's two sides of the same coin. Is a vaccine also? I have to say that also working on the immune mediators of vaccine progression in HIV-infected individuals will provide very important information for the development of therapeutic vaccines, a topic that we didn't cover today.
Okay, thank you. One last, you've touched on broadly neutralizing antibodies, but the question that was submitted is, what is the progress with broadly neutralizing antibodies trials in humans? Is there anything else you'd wanted to add?
Well, I mean,
to your comments?
Sure. I mean, as I mentioned, the groups of Dennis Burton and Bart Haynes are exploring in preclinical work how to induce broadly neutralizing antibodies by sequential immunizations with antigens that have different level of mutation. I mean, vaccinating first with antigens that combine to jump cell line, and then basically guiding the maturation of the immune response by exposing the volunteer to different antigens along the maturation pathway. Now, that has been tested in animal models, but not in humans yet. So my feeling is that I would like to see some of these very exciting new concepts moving to clinical trials sooner rather than later, because your germ lines in humans are different from monkeys. We have very similar immune responses, but they are immune system, but they are different. So far there have not been a lot of recent work on that. But remember, the first wave of HIV vaccines were aimed at inducing neutralizing antibodies. And going back to some of those early trials and trying to understand what happened with the new knowledge, with new tools. For instance, very recently was reported that pediatric vaccine trials that were conducted a few years ago and were declared to be failures because they didn't induce any interesting immune response. When the samples were reanalyzed, they found that some of those trials some of these vaccines were actually inducing the potentially protective immune response that were identified in RB144. So I am a very strong believer that we need to do quickly human trials and do not preclinical, clinical, but paraclinical research. So it's in parallel animal and human studies. Of course, human studies have to be done with the highest ethical standards, of course.
Okay, and your presentation did cover this, but the question came in is, what is the current stage of HIV vaccine development internationally, and how has the environment or context changed over the last 5 years?
Well, internationally, there have been some changes. I mean, I have to say this is a very personal opinion. I don't work with any organization now. I'm free to have my personal opinion. Thailand, for instance, has been a country where most of the trials have been conducted, including the only efficacy trial that show efficacy, RB144. Now, there is a lot of effort in conducting additional efficacy trials in Africa, and I totally understand the reasons. I mean, this is where the epidemic is more severe. However, the point I would like to make is that we need a vaccine that is globally relevant, a vaccine that can protect people in Thailand, people in Africa, people in North America, in South America. And we need to develop a strategy. I have some ideas that this is not the place to discuss, but we need to have some strategy to answer those critical questions in parallel. I mean, I don't have patience, not the age, to wait for an efficacy trial to show to show protection in South Africa, then to think about how to extend that information to other areas of the world. I think that we need to approach the problem in a more coherent way in integrating new knowledge. And I mentioned already that the mosaic vaccine developed by Dan Baruch may be one approach, but not the only one, but one approach for the development of what we need is a globally relevant vaccine. The other problem, just give me 1 minute more, is funding. Funding is decreasing. Funding for HIV vaccine is going down. We have not seen, Canada is an exception, is a great exception, where Canada has put money, their own money, funds, to support HIV vaccine research. We don't see that in many other countries. When the enterprise was established, our hope, our expectation was that other countries, particularly European countries, will contribute significantly to the global effort to develop an HIV vaccine. That is not happening. I'm concerned that the society somehow has led to believe that we already have the tools to control the epidemic, and maybe that an HIV vaccine is not needed after all. But one of my last slides was precisely to make the point that unless we have an HIV vaccine, we will not be able to control the epidemic. In the United States, in the country where I live now, there are every year 50,000 new HIV infections every year. And that number of 50,000 has not changed in the last 10 years. So don't tell me that we have the tools to prevent the epidemic, because not even in this country, those tools have been able to decrease the number of new HIV infections to a very low level.
Okay, our next question, thank you, is what do you think the role of researcher funders is in funding the outside the paradigm science? Would you have any recommendations for researcher funders in this regard?
Well, researchers is to ask questions that are not been asked, not to become just a Me Too kind of scientist. Funders have the major responsibility. Funders have to allocate a proportion of the funds. I suggested 10% of the funds to really out of the paradigm research. Now, I didn't have time to discuss the models, but there are several models. I mean, I know, for instance, the NIH has explored different approaches to fund innovative research. The Gates Foundation, especially with the Grand Challenge Exploration, also provides a model for funding out of the paradigm research. What I can advance is that I believe that the standard peer review mechanism is not appropriate for innovative research. Now, in the Gates Foundation, when I was there, we used the champion approach. So if there was a panel of reviewers, and if one member of the committee became a champion of a crazy idea. That crazy idea was supported. But the problem with innovative research is that it has three major problems. One is that they doesn't have pre-existing data because it's new. I mean, it's not the same that with current science, where scientists come with a lot of data to support their next grant. With innovative ideas, we don't have the data. Second, with innovative ideas, we don't have peers. When an idea is new, there are not many people in the community that can be considered peers of that review, of that project. That's why peer review doesn't work. And the third reason is that, unfortunately, but that's the reality that we have to confront, is that most innovative ideas actually fail. So funding innovation is typically difficult. It's not trivial.
Okay, thank you. Just two more questions. We have one. If we are seeking novel paradigms against a mucosally transmitted infection that targets the mucosa, why don't we have a strong effort focused on mucosal HIV vaccines?
Yes. Why not? We don't know. I don't know. In fact, there is only, if I remember correctly, maybe there's only one group that has actually conducted HIV vaccine, mucosal vaccines. And this is Robin Shattuck in London. I know Canada has is a large amount of experts in the area. So I believe that mucosal immunization approaches to prevent infection at the mucosal level need to be stepped up. Now, you remember that with other diseases like hepatitis B, for instance, you don't need actually to use mucosal immunization to prevent mucosal transmission. You can do it by systemic antibodies just translate into the mucosal level. So, but there is some work. There is some work on mucosal immunity, and I feel that it should increase.
Okay, last question. What is the possible role of the GI lymphoid system in immunity?
Oh God, I'm a virologist, not an immunologist. So I'm not sure I can give a credible answer to the question. I'm sorry.
Okay. All right then. Okay. So if I don't see any other questions coming up in the chat box and no other signs, so we'll cut off the questions for now. I'd like to thank you, Dr. Esparza, for your wonderful presentation and for taking the time to be here with us today. Thank you, everyone, for their participation and in this fruitful question and answer period in our webinar, Do We Need a New Paradigm for HIV Vaccine Development? And I'd like to thank you, Jose, also for sharing your expertise with the members of the CHVI Alliance virtual community. I'd also like to thank Elizabeth Hanless, our instructional design consultant, for her work in facilitating this webinar, and remind everyone to complete the webinar evaluation using the link on the screen to let us know your thoughts about the webinar. I will also remind everyone that the webinar has been recorded and will be made available in the training tab within the Alliance virtual community, and watch for announcements about its availability on this discussion forum of the virtual community. So once again, thank you so much, Jose, for taking the time to be with us today.
Thank you very much.